Okay, now we're going to talk about the actual neurons, the sensory neurons that respond to the light energy that is making its way, remember, through your cornea, through the pupil, through the lens, and then is being, typically you're trying to focus that information on the fovea. Remember that sort of pitted area of the retina at the back of the eye, and the cells that are going to carry out what we call phototransduction, right, are actually called photoreceptors, which makes good sense, right? And it turns out that typically we have about 4 different types of photoreceptors. There are 2 classes we refer to, but one of the classes contains 3 distinct photoreceptors, and in some cases, I'll tell you, there is a 4th. So this class of photoreceptor is referred to as a cone photoreceptor. These are the cone photoreceptors, and if you look back here, this is the back of that cell at the back of your retina. There's a series of membranous discs actually here where you will find what's called a photopigment. This is a special protein that is generated, from instructions in your genes, and this particular cone will express a photopigment that will be able to absorb, a particular wavelength of light, right? And the cones, the 3 cones that I'm suggesting most of us have, actually are distinguished by the type of photopigment that they express. So you have red cone cells, right, that actually express a pigment that selectively absorbs and can phototransduce red light, right? And you have blue cone photoreceptors that selectively absorb light in the blue wavelength, and you also have green photoreceptors that express a different pigment protein that's found back here in these membranous discs that correlates with your ultimate perception of green light. If you lack one of these cone photoreceptors, or the pigment's not functional in one of them, you may have some sort of color blindness, right? And on the flip side, they found that in some women, there's a 4th cone photoreceptor that's been identified with a 4th photopigment, and that photopigment absorbs a slightly different wavelength of light, so it may confer greater kind of color discriminative capabilities on some women. Now, the other type of photoreceptor is referred to as a rod photoreceptor. That's this one over here, and you should note that the pigment, the location of the pigment in the rod cell is found back here in this very long rod-shaped series of membranous discs. So there's definitely a distinct difference, structural difference, right, between the cones and the rods. The pigment also that the rod expresses is far less discriminative. It just responds to a wider range of visible light. A wider range of visible light will be absorbed by that photopigment and provoke a response. The other thing you should note is that the cones don't have too many membranous discs, right? There isn't a big kind of receptive region on a cone, certainly in comparison to the rod, which is a much bigger receptive, kind of area for the capture and absorption, right, of particular wavelengths of light. So it turns out that the cones are far more active when there is a lot more light. When you, for example, are in daylight, right, or there's a light on in a room, there's enough photons, there's enough light energy sort of coursing around, electromagnetic radiation, within that visible light spectrum, that the cones can pick it up, can actually detect it. While when the light drops, right, when dusk comes, for example, you're more at the mercy of the rod cells, which don't have the same color discriminative capabilities because there's only one kind, there's only one pigment, right? So at night, or at dusk, your color discriminative capabilities drop significantly, right? The other thing to know about rods and cones is that that area of the retina that I was referring to, where you have the ganglion cells and the bipolar cells sort of pulled aside. This is where you're trying to focus when you're trying to you get the most information out of some area you're examining visually. That foveal region, that's where you find a lot of very tightly packed cone cells. And the other thing to know about this very tight packing of cone cells in this foveal region is that they are linked. Here's the the synapse with the bipolar cell, right? This is, remember, this is from outside the eye, right? This is the light coming in the the cornea, and then going through the pupil, and then the lens, right, then working its way through the vitreous humor, and then it's going to go through some of the cells ahead here, and then it's going to go be captured by the pigment found on the membranous disc back here in the cone. Well, then it's going to provoke some kind of neuronal response, and this is going to be communicated here to a bipolar cell, right, which is that next layer up, and then to a ganglion cell, and then you're going to have the ganglion cell axon leave the eye, right? There's a lot of one-to-one-to-one mapping of information and transfer of information at the level of the fovea. Very tightly packed cone cells, and they are linked, each one to one bipolar cell, then to one ganglion cell, and that information goes out. In contrast, when you start getting more to the periphery, away from the fovea, right, off to the side of your vision, right, you have more of these rod cells, and multiple rod cells, it turns out, are linked up to like a single bipolar cell, which is then linked up to a single ganglion cell. So those ganglion cells are actually transmitting information from a wider range. It's not as detailed in terms of pixelation, right, as it is at the fovea. The fovea, you have very tightly packed cone cells, you get high pixel level, you have very detailed fine vision and color vision, and it's also best, remember, with so few of these membranous discs and so little pigment, it's best in good lighting conditions, while with the rod cells, which are more in the periphery, and many of them are linked up to a single bipolar cell, which then goes on a ganglion cell, that's carrying information across a slightly broader range, right, of visual space. And you can tell this, actually, if you think about it, like if you, let's say it's at night and you're trying to look at a star right up in the sky, right? If you try to foveate it, try to focus it on your fovea, all of a sudden it can just disappear. You can't see it. There's not enough light, coming from that star to activate your cone cells. However, you look off to the side, you actually get a better impression of the star. You can see it. The other thing to think about is because these rod cells in the periphery are linked, multiple ones are linked together to one bipolar cell to one ganglion cell, they're better at detecting something that's moving across several of these cones. So you have better detection of sort of movement, things that are moving off to the periphery as well, which is also very useful from an evolutionary standpoint. Cheers.